Good to have you on Planetterrian Daily, episode 57, coming to you on May 8th, 2026. Here's your science and health briefing. Macrophage volume shifts trigger immune danger signals in new cell biology findings. A new study in the Journal of Cell Biology shows that changes in macrophage cell volume alone can trigger immune activation and inflammation. This happens without depending entirely on surface receptor detection of pathogens or damage signals. The volume shifts happen quickly through ion channels and adjustments in cytoskeletal tension, often within seconds to minutes. The work reveals an internal sensing system that allows these immune cells to respond to physical stress or osmotic changes directly. Such a pathway could help explain why chronic inflammatory conditions persist even when traditional receptor signals are absent or blocked. Targeting volume-sensitive mechanisms might offer new ways to calm overactive immune responses in diseases where inflammation lingers. The finding also suggests that everyday factors like tissue swelling or fluid shifts could influence immune readiness more than previously recognized. The Planetterrian Spotlight this week focuses on that same macrophage volume sensing mechanism. The discovery clarifies an elusive early step in how the innate immune system registers trouble and begins mounting inflammation. Because volume regulation operates across many cell types, the results may reshape thinking about inflammatory disorders that arise when this internal check fails. It also opens possibilities for modulating immune activity by acting on volume pathways instead of surface receptors alone. This internal monitoring could turn out to be a more universal alert system than receptor binding in certain settings. Researchers are now considering how disruptions in cell volume might link to broader patterns of immune dysfunction seen in aging or metabolic stress. The spotlight leaves open the question of which other immune sensing routes might operate through similar physical changes inside cells. Now, here's something most people get wrong about how immune cells detect problems. Most assume macrophages rely only on surface receptors to spot pathogens or tissue damage. In reality, these cells also track their own internal volume as a fast, early warning system. Right now, as you listen, your macrophages are adjusting ion channels and cytoskeletal tension continuously. Any abrupt swelling or shrinkage can switch them into alert mode and prompt release of inflammatory signals. These volume changes unfold on a timescale of seconds to minutes, which is quicker than many receptor-driven reactions. The practical takeaway is that therapies aimed at chronic inflammation could someday restore normal volume regulation inside cells, rather than only blocking later cytokine steps. Watch for follow-up work testing whether common issues like edema or osmotic stress change infection susceptibility through this same route. On a different note, researchers have identified a fresh way to activate the STING pathway that could strengthen immune recognition of tumors. The work published in Science magazine describes a new mechanism that engages STING to improve anti-cancer immunity. This approach broadens the molecular targets available for future immunotherapy strategies beyond current agonists. The findings remain at the mechanistic level for now, yet they increase the options for designing treatments that help the immune system spot and attack cancer cells more effectively. The discovery matters because it shows there are additional entry points into the pathway that had not been fully mapped before. It also suggests that combining this route with existing immunotherapies might produce stronger or more durable responses in certain cancers. The research keeps the focus on basic molecular immunology while expanding what is possible in the lab. Next up, a large-scale look at publication patterns reveals how scientific careers evolve over time. Analysis in Science magazine of many papers shows that longer careers shift output toward incremental combinations of existing ideas rather than disruptive new advances. The pattern holds across multiple fields and appears tied to structural aspects of how research careers progress. As scientists accumulate experience and responsibilities, their work tends to synthesize prior findings more than it overturns established views. This trend raises questions about whether current incentives in academia and funding favor steady accumulation over high-risk novelty. The study does not judge the value of either style, but highlights how career length itself influences the balance between synthesis and breakthrough. Understanding this dynamic could inform how institutions support different phases of a researcher's productivity. Finally, a recent Lancet cover story draws attention to a case where AI-generated fake references made it through peer review. The fabricated references appeared in a published paper and illustrate the growing difficulty of spotting content assisted or created by language models. The incident serves as a reminder that research integrity checks must adapt to new tools that can produce convincing but false citations. Peer review processes now face added pressure to verify not only data and conclusions, but also the authenticity of every listed source. The case does not suggest widespread fraud, but shows how even careful review can miss AI-assisted manipulation at present. Journals and reviewers are likely to refine their methods in response, which may slow some workflows while improving overall reliability. Before we go, keep an eye on follow-up studies that test whether volume sensing pathways in macrophages extend to other immune or tissue cells under everyday stress conditions. That covers today's science and health news. Share this with someone who's curious about the latest research. I'm Patrick in Vancouver. See you next time. This episode used AI voice synthesis of my voice. Editorial selection and analysis are my own.